RNA. And even before we got to RNA, we really studied in really great detail the structure of DNA. I'm comfortable with looking at your handouts that you guys brought in, that you're understanding much better the structure of DNA, say, than what you had knowledge of, say, a month ago. Okay? You understand that there's carbons that can give us location, etc. You also learn that we can apply these same concepts to another similar molecule called RNA. And what we learned is there are different subtypes of RNA. What are the three subtypes of RNA? Messenger RNA, transfer RNA, and ribosomal RNA. They're all types of RNA. They all have adenine, cytosine, guanine, or uracil bases. The sequence obviously is going to vary between the different types, as well as the length. We also learned that the structure of them is different because they all what differently? They're built different, so they do different things. They work different. They function differently. Okay? And so what is the function of messenger RNA? It doesn't read the codons. It actually, what? Yes, I'm going to say technical clarification on your term. The messenger RNA is actually, it does carry the information to read DNA codes. It codes for the protein, amino acids. Okay. So let's restate that. We had a lot of answers coming in that all could be fairly accurate. Okay. So messenger RNA, just think of the name. It carries the what? The message. What is it messaging for? What are we trying to ultimately make? Protein assembly. Very good. So the messenger RNA codes for the message, or the sequence of amino acids. Where we'll apply this is during translation, is that it is arranged in what's called codons. Our messenger RNA is arranged in codons when we start to using it. And what's a codon? It's just the code of how many? Three what? It's just a sequence of three, the bases. It's just a sequence of three RNA bases that code for a single what? Amino acid. Very good. It's just a sequence of three of those mRNA molecules in a row. I guess nucleotides would be best to say. Three of those in a row that code for a single amino acid. Right now we're just putting the definition out there. We'll conclude the class today with actually practicing it and see what the heck that means. It'll make more sense to you when we do this work. Okay? So the messenger RNA codes for individual amino acids because of the way its RNA molecules are arranged. And what about transfer RNA? It has the anticodon, the very good. The transfer RNA, or the tRNA, one part of it has an anticodon, which starts to get a little crazy, but that just means what? An anticodon is the what? Very good. It's the complement of the codon. Get used to the term. The transfer RNA is just the complement of the mRNA codon. Just has the ability to bind mRNA, basically. It gives us all that anticodon does is give the ability to the transfer RNA to bind to a specific sequence of mRNA. Because why? Why do we need that specific binding? What other feature of transfer RNA besides expressing an anticodon? Very good. It binds to a specific amino acid. That anticodon in the transfer RNA we notice the transfer RNA also binds to a specific amino acid. We haven't gone through the different amino acids, but we know from the first unit exam material that amino acids are building blocks or are monomers for proteins. So the transfer RNA is just kind of our intermediate communicator. I don't know if that's the right word to use, but as the name implies, it transfers a specific amino acid based on its complement, the transfer RNA's complement binding with the mRNA codon. So the takeaway before looking at it in the process at this point is that the transfer RNA has two areas. One is called the what? It binds to the message. So what part of the what's the name of the transfer RNA area that binds to the messenger RNA? The anticodon. Okay. So transfer RNA has a mRNA binding area called an anticodon, and the other area that it binds to an amino acid. Very good. We also noted the transfer RNA structure is pretty unique in that it folds so those two ends are exposed to bind the anticodon area as well as the amino acid binding area. The third type of RNA that we discussed is called ribosomal RNA. It's aptly named because it's found in this big structure called a what? A ribosome. Okay, it's found inside of this structure called a ribosome. The ribosomal RNA is probably the hardest to kind of conceptualize because it's not just RNA, it's actually in a protein. Okay, so it's just a big macromolecule. We'll learn that the role of the ribosomal RNA is to work in the ribosome to help assemble our growing peptides or our growing proteins. Okay, so it's going to be where the bonds, the peptide bonds are actually formed when we look at this process called translation. So if we summarize the basic structure of these things, which RNA is this represented in this image? How do you know it's messenger RNA? It's single-stranded, and that strand is arranged how? Straight. Okay. In this mRNA molecule, in a printout, you'll always see it written 5', 3', that gives the directionality. But remember, this is a three-dimensional molecule, so your cells don't know reading left to right or whatever. There'll be recognition particles that recognize the certain regions. Okay. So we know this is messenger RNA because it's single-stranded and it's linear, you guys call straight. If I were to go through and try to find the codons in the light green area, don't look at the darker green area. What is the sequence of nucleotides in this first codon? What would the nucleotides for the first codon be? So don't look at this green part. The first codon sequence would be A, U, G. I'm going to read in triplets. The first sequence I'm going to block this off. There always has to be a kind of a binding, getting ready site. All right, so I'm not going to address that yet. But if we start reading this in sequence, if I read these in triplicates, I just highlighted a single what? Codon. And the next thing would GGA would be another codon. UGA would be another codon. I could just keep going on. There's a question? Yeah, why is that? It's a leader sequence. I just really want you guys to understand the basics before getting into some details, and that's a detail something. I don't know that we're going to, we're in our third day now. I was hoping to finish this up last week, so I may not. It's called the leader sequence. It initiates the binding of the ribosomes, but it doesn't have any coding action. So we want to talk about the coding of the proteins. back of the room was, well, we can kind of begin to understand how the messenger RNA is read, R-E-A-D, in triplets, or called codons. The question was, well, is there anything that dictates the number of codons? Yes, there is something that dictates that, and that's the DNA. Okay? Not all proteins are built the same. Some proteins may be technically just what's called a tripeptide, and they're just 3 amino acids in length. So how many mRNA nucleotides would there be? There's 3 codons, so how many? 3 times 3 would be 9. Okay? Some of them are hundreds of amino acids in length. Okay? So it just depends on the ultimate structure of the protein. Bigger proteins will need a larger code. Smaller proteins would need a smaller code. But not all mRNA messages are the same length, because not all proteins are the same length. There was a second question?
Isn't AUG a starter codon?
Yes. Okay, and you are on it. What we're going to learn, I think it's, I don't remember the table reference, but when we get into translation, AUG is the actual, what we call true start codon. If you see the sequence of nucleotides, it codes for an amino acid called methionine. And if it's right behind that leader sequence, that dark green area you guys had a question about, that initiates the start of that protein sequence.
Is it always a starter codon, or would it vary between different?
No. AUG is the common as far as we know. Like, there's not been anything that otherwise support that. That's been the accepted start codon. But, right, there's some cool research going on to say, well, that's where we interpret everything. That's how we analyze everything. We've just been taking that as face value. So if we take it at face value, the short answer is it's always AUG. However, there's some scientists trying to say, well, that may not be the case. And so that may be falsified in the next 20 years or so. So that's brand new kind of thought process. Good question. So AUG, and any time in this classroom, we'll always kind of start at the AUG start a sequence, and I'll give them to you in threes, right? So there's nothing trickery about trying to figure out. But if you're a scientist and you go sequence some messenger RNA, you got to figure out is your sequence coming out 5' 3' or 3' 5'? You've got to figure out where the leader sequence ends. I mean, there's a lot of interpretation, so it's not as cut and dry as these images might actually imply.
There were some of these on the homework, when they weren't smart,
Okay.
and they were different letters than so I really just guessing about what the
So,
I wasn't sure how to do it.
depends on which process you're looking at. If you're talking about transcription, if you're trying to take a DNA sequence and make an mRNA sequence, that's when you do that complement with the uracil substitution for the thymine.
Right,
Okay?
and I was looking for that, but it has different letters, specifically with an H in there.
If you see H's and L's and Y's, that's the amino acid code. Yeah, I'm not going to have you learn all the different amino acids. Okay. So if you see letters other than A, U, G, C, okay, am I missing something? Or T, that is not a nucleic acid. That's actually an amino acid shorthand note. Okay. And so from that, you could work backwards from a protein to an RNA to a DNA sequence. All coded for. All right, so messenger RNA, linear. We'll read it 5' to 3', and we always look for the what? So I want to figure out what it codes for. I first look for the starting codon, which has a sequence of AUG. Very good. Which amino acid is, or excuse me, which RNA is represented in the second image? Transfer RNA. How did you know it's transfer RNA? One, we can see an anticodon. Two, we can see it's folded. What else? Kind of L-shaped. It is single-stranded. I didn't say that, but it is folded. Single-stranded and folded upon itself. what kind of connectors for? So we have the anticodon, and also another area bound with something, an amino acid. The third type of RNA is called ribosomal RNA, which is embedded inside of this big protein, making up that whole thing that we see in this third image is actually the collective ribosome. I think the little blue, I can't differentiate in there, but embedded, internalized would be little single-stranded segments of the RNA, right? But collectively, that whole thing is a ribosome itself. If we look at all three types of our RNA molecules, we might have talked about this. We talked about this on Thursday. Alright. So all types of RNA are manufactured during transcription, not just messenger RNA. We tend to hone in on messenger RNA because that's one that codes for our message. It's really, really important for the production of a protein. So all of our RNA molecules are manufactured during transcription, which happens where? In the nucleus. Okay? Good. All of our RNA molecules, whether ribosomal, messenger, or transfer, they all have a 5' and a 3' end. Additionally, your book goes into the modification, but I'm going to spare you, okay? And we're not going to go into the different modifications of the different types of RNA molecules. So what that means is, how does that mRNA get ready, right? It undergoes specific additions to the different ends and splicing and all kinds of really cool stuff, as well as the ribosomal RNA and the transfer RNA, right? They've got to get charged with the amino acid, etc. So they don't magically just kind of come out ready to go. They get processed, okay? Following transcription. And what we'll find is each of these RNA molecules will have a specific role during translation, okay? Or what I have listed here as during protein synthesis. They're all going to work together during translation. Any questions about the specific types of RNA molecules before we see them made and then used? I want to review the phases of transcription and translation, which you actually addressed when you did your worksheets that you submitted last Thursday. So you have a kind of a primer, and we'll just look at it in a little more technical detail. I want to let you know, though, whether you're looking at transcription or translation, I tried to give you a heads up a couple of weeks ago that the names are really extremely similar. So if you're just looking at them for the first time, it's so easy to get them confused. So it's imperative that you practice writing what the words mean and just keep repeating it until it just becomes kind of inherent where you can use it in a dialogue accurately. So one, just the name sounds similar. Additionally, the three subphases of each of these processes are the exact same. You initiate the process, you continue it, and you stop it. Okay? And so that also allows for some similarity and therefore potential confusion. However, we know that the processes occur in different locations and manufacture different end products. Transcription, you just identified, happens inside of the nucleus to make what? What are we making during transcription? Different types of RNA, whether it be messenger or transfer or ribosomal RNA. Once that RNA is manufactured, the next process called what can occur? Translation. Which in a eukaryotic cell will happen where? In the this cytoplasm, and some of you guys also identified accurately in your handout, it may occur in the rough ER if the protein is to be secreted. So if the protein is to be secreted, remember the role of the rough ER is to make proteins and secrete them, so it could occur at the rough ER or it could occur in the cytoplasm. So you guys had that both on your worksheet that you turned in last week. But the whole point of it is to manufacture what? Translation manufactures a protein. The different types of proteins. If we look at the stages of transcription, I mentioned there are just 3 phases: initiation, elongation, and termination. Okay? During initiation, obviously we're starting the process, as the name implies, right? What are we trying to manufacture again during transcription? RNA. And so I'm trying to make a molecule, trying to carry out a metabolic process, so I probably need to use something, as illustrated by the purple blob, a type of what? Energy is utilized. A protein that's functioning as an carrier, transfer, speeding up chemical reactions. Enzyme. Very good. Okay. It's an enzymatically regulated process. Transcription, just like every other process that we'll study, utilizes specific enzymes. We are making RNA, right? So what is the name of the enzyme used to synthesize polymerase? And more specifically, the prefix, we could say, what kind of polymerase? RNA polymerase. So, an enzyme called RNA polymerase, I'll write this on the board for you. RNA polymerase. If you work through the word backwards, ASE tells you it's a what? Enzyme. It has something to do with manufacturing a polymer, and more specifically, which polymer? RNA. It's an enzyme used to make, or different types of RNA. Because we have different types of RNA, there's actually different types of RNA polymerases. And we'll just call it RNA polymerase. Manufactures RNA molecules in a cell's what? Nucleus. Okay. Before I get too deep into this process, we're trying to make RNA to eventually, during translation, manufacture individual what? Proteins. The whole concept of this is to make a single type of protein to function inside of that cell or message or something like that. Okay, so keep that in mind. This whole process is just make a specific type of protein. Okay? In order to do so, we first have to get the copy or the recipe from the DNA. So we don't want to damage the DNA. What color is the DNA represented as in this image? The blue. What is the purple thing? The enzyme called RNA polymerase. First thing is, during initiation, RNA polymerase actually has to bind to what? To what? That's really, yes, of the what? Promoter region of just the blue stuff called DNA. First thing is, the RNA polymerase has to bind to the DNA. And she's correct. It'll bind to an area called the promoter. Okay, so write this word down. RNA polymerase binds to an area of the DNA called the promoter. The promoter is a region of DNA that's found where? In respect to the gene that's going to be eventually expressed. Well, where? The beginning, before it. Okay? Before. Okay? So the promoter region is the area where RNA polymerase binds right before the gene code is found. The promoter region of DNA is the proper term is called upstream. Here's a part of the DNA sequence before the actual code, the gene code itself. Why is it important that the RNA polymerase binds upstream the gene? It signals the gene start. If I bind it in the middle of the gene, would I get a full copy of the gene? No. If I bind it at the end, I wouldn't get any copy of the gene. So it's important that the process starts before the code. And that's called the what region? Promoter region. What else does RNA polymerase do during initiation besides just binding to the promoter region? What can we see in this image? It's not damaging the DNA, but it is separating the strands of DNA. What is it separating? It is exposing what's called the template strand. What did we have to break here to expose the template strand? Hydrogen bonds. So RNA polymerase breaks the hydrogen bonds between the two strands, and now our nucleotides are exposed. No, just don't go there yet. Just first thing is RNA polymerase not only binds to the promoter region, it does what to those hydrogen bonds between the strands? Separates them. Once those hydrogen bonds are broken, what did we say is exposed that can be used? The DNA, and it is called the template. That's what's going to be read, R-E-A-D, the template of the DNA. It just means one strand of DNA is exposed and it's going to be used. So during initiation, RNA polymerase binds what's called upstream to a gene at the promoter region. Then it breaks the hydrogen bonds to expose the gene template. That just initiates it. Then we don't need to just open it up. We need to actually copy it complementarily, right, using our RNA nucleotide. That happens during what's called the elongation process. During elongation, our messenger, or you can just say our RNA molecule, is synthesized complementarily to what? The RNA's complement to the DNA template. question is, well, how long is the RNA and mRNA molecule? Well, it just depends on how long the gene is. Okay? This elongation complementarily process continues. It's powered by RNA polymerase. And then at some point, it's got to stop. Okay? It's not an infinite process. When it stops, the process is called what? Termination. Okay? There are many ways in which termination can occur. Your book may even go into more technical details. If you guys at this point can just say it stops, it terminates, what happens during termination? The mRNA is no longer being built and it separates from the what? From the DNA and the polymerase. Polymerase lets go of everything. The RNA is released. What happens to polymerase? It's also released. What about the DNA? It goes back to its normal shape. So in other words, those hydrogen bonds reform, the helical nature it retwists, and it may refold back into a chromatin or something like that. It refolds to protect itself. Would the DNA have been altered in any fashion? Like permanently altered in any fashion? No. We don't want to damage the DNA, right? We want it to go back to its original state so we can reuse it later when necessary. This process is extremely quick. Within a few, right, within less than a minute or something. Depends on the length of the gene. If you're looking at a prokaryotic versus a eukaryotic organism, but within just a few seconds, and if you look at over a period of a few minutes, you can get a lot of transcript or RNA product actually synthesized. It's very fast because it's utilizing enzymes. We also don't want to kind of handle the DNA too long, so we're just kind of quick, get it over with. The stages of transcription are summarized for you in figure 711, which we kind of already walked through. So before I move on, I want to address a couple of questions you guys had about the promoter region. Where is the promoter region found? Before the coding regions, good, of the gene or the DNA. If I talk about the DNA template, that's the template that's what? Used, read to manufacture RNA. But yes, it has to be separated before we can use it. Very good. And the enzyme that produces RNA is called RNA polymerase. After that, we can manufacture an RNA molecule. In this case, probably a messenger RNA. Usually in these textbooks, we focus on the messenger RNA. We can see the 5' 3' directionality and the complement arrangement between RNA and DNA as well. What color is the RNA molecule in these images? That kind of green color structure. This will continue until some part of the gene says to stop, okay? And that initiates a process called termination, where this whole transcriptional unit will just disassemble. The DNA will resume its original shape, and the RNA can go on and be modified and eventually utilized, right, inside of the cell. So if we were to summarize transcription, where does it occur? We said it occurs in the nucleus of eukaryotic organisms. What about prokaryotes? Where would it occur? Cytoplasm. Or if you remember, our nucleus of prokaryotic organisms is called the nucleoid region. So technically there, but it's not membrane bound. So transcription occurs in eukaryotic cells Nucleus, because that's where it has all of its what? DNA. Bless you. Which molecules are utilized? We definitely need DNA. We also need it's not used per se, but it's definitely what? RNA is definitely made. Okay? So the whole point of transcription is to manufacture RNA. So let's go back. What else is used besides DNA to do this process? We also need a molecule called an enzyme called RNA polymerase. Very good. Questions at this point you have about transcription? Yes.
In the book, it says that elongation is the nucleotides are added to the 3 end.
Yeah, it grows in that 3' end.
Right.
The RNA is manufactured 5' and 3'. It's manufactured kind of in order, if you will. So it goes out and it's ready to go something. Okay. So if you want to add that, the RNA is manufactured 5' to 3'. It's made, synthesized, polymerized, kind of in order. You might read it. Additional questions about transcription? Even if transcription occurs, that's not the end of the story, because really we're trying to make a protein. Have we made a protein yet? No. So we need to follow through with a process called what? Translation. And let's contrast translation. Should we give you transcription translation on yesterday or Thursday? Alright, so we're going to go to the next phase of translation, which we're going to utilize all 3 types of the RNA molecules, messenger, transfer, and ribosomal RNA to manufacture a protein. As I mentioned, the names are similar as well as the main stages, so we're going to still have initiation, elongation, and termination. However, we're making a completely different molecule in this process. During translation, the process is initiated, where if you remember, we said we're going to utilize all 3 types of RNA molecules. The one that codes is called what? Messenger RNA. Okay, so during initiation, messenger RNA is available. It's not manufactured. Messenger RNA during initiation will bind to the small part, or it's called the small ribosomal subunit, small subunit of a ribosome. Okay, so during initiation, a small subunit of a ribosome will bind to a messenger RNA molecule that just got transcribed. In this image, you can kind of see the small ribosomal subunits. This little kind of green blob. And that's the ribosome. You can see the messenger RNA in the gray kind of scale linear strand. We can also begin to see transfer RNAs begin to bind complementarily. If you look at this oversimplified image, the purple thing is a what? An amino acid. It must be bound to what type of RNA? Transfer RNA, which we can see really enlarged. The transfer RNA is what in dark green? It's what? Anticodon. binding complementarily to the RNA's, the messenger RNA's what? Anticodons bind with codons. Very good. Which amino acid do you think is going to be the first one to bind? I mentioned it briefly, you may or may not have caught it. You mentioned it earlier called methionine, MET. So the first codon, right, or first amino acid is brought into order. Messenger RNA binds with a small ribosomal subunit in the first transfer RNA carrying which amino acid? Methionine amino acid. This unit isn't fully functional until the other component of the ribosome assembles, and so the large ribosomal subunit has to bind. This is a three-dimensional interlacing of molecules, and so these images in your textbook are just cross-sections so you can kind of peekaboo inside of there. These are big kind of globular molecules. So during initiation, messenger RNA binds with the what? Small ribosomal subunit, as well as transfer RNA carrying the amino acid methionine. Then, once that's ready, what can come in and bind? The large ribosomal subunit. That translational machinery is ready to go to work after we initiate it. We can then elongate what's called the growing polypeptide chain, or basically make a protein. The mRNA is, if you will, pulled through the ribosome, one codon at a time. It's reading three nucleotides at a time, or a codon. As it's assembled, there's enough transfer RNA and amino acids available that the right transfer RNA will come in and bind complementarily to the codon in sequence, bringing with them a new type of what? Amino acid. Just because the amino acids are there, though, those amino acids then have to be what? Bound. So during elongation, the mRNA is read in codons. And amino acids are brought in by the transfer RNA. As well as those amino acids are bound together. Remember the type of bond that holds our amino acids together? It's a covalent, is a peptide bond. More specifically, it's a peptide bond, but certainly it is a covalent. So our peptide bond is formed between individual amino acids in the middle of this, during the middle of this process. Transfer RNA. They're transferring the amino acids. If you look at the image summarized from your textbook, you can see the transfer RNA binding anticodon to the codon. You can see the triplets, or can you see the triplets? You can also see that it's bound to the amino acids. If you look in this middle image, you can see the first amino acid, and the purple line is representing that peptide bond, bound to another amino acid, which is forming a peptide bond with the third amino acid. And it just continues building these peptide bonds during the elongation process. So that full translational machinery, the full ribosome put together, reading the RNA, the messenger RNA and codons, and the transfer RNA bringing in the correct amino acids based on the code. Peptide bond forming. Eventually, we're going to get to where? Termination, or the end. Okay. During termination, what do you think happens? Elongation stops because we reach what type of codon? A stop codon. During termination, a stop codon is reached. can enter in there? No amino acids, no more transfer RNAs. They can enter. Those transfer RNAs, the machinery runs off of the messenger RNA. Transfer RNAs go out, the amino acids dissociate. And we have built a what? We've built a protein. The manufacturing of a protein, or the synthesis of a protein. Let's go through and just look at the larger images of these phases, then I'll address questions. Okay. So during initiation, okay, first thing is, how can I tell this is translation versus transcription? How can I tell that this is not transcription? What is apparent here? The ribosome. Okay, what else did you guys say? The amino acid as well. The ribosome's present, the amino acids are present. We can see the transfer RNA working with the messenger RNA. Okay, so that tells me this is translation. If I see DNA in the RNA molecule, that process is called transcription. So we're in translation where we're trying to make a protein. Okay, so we went through the phases of initiation where the messenger RNA binds to a small ribosomal subunit. That first transfer RNA comes in, and then large subunit joins the complex. It's very fast. I mean, we're taking minutes to describe it. It just takes portions of a second to actually carry forward. And the way that ribosome works is it has catalytic ability. So it's just kind of pulling that mRNA through, it's reading it, and just letting those transfer RNAs come in and add amino acids and join those amino acids together. Okay, so there's a lot going on during the elongation process of translation. This will continue until the mRNA codes for a what? Stop, okay, or termination, or a stop codon. We'll just continue on, and then it will terminate. And here's really what we're kind of focusing on. Yes, the RNA is very important, but here's the end product. End product is an amino acid, right, or a bunch of amino acids joined together to form a protein. This is the question that you may have had a moment ago. What are the, sometimes you see the letters like M or L or R or something like that. If you see the rest of the alphabet, that's actually representing what type of let's see, an R, an L, A, amino acid. So the whole point of this process is to join amino acids to form proteins. And so if we were to summarize this, well, I'll come back to this image. This is important, so I want to spend more time on it. But if we were to summarize translation, where does it take place? Takes place in the cytoplasm, or maybe on the rough ER. But not in the nucleus.
Oh, that's the other one.
Alright, so translation does take place in the cytoplasm, or you could say in the rough ER. As long as you acknowledge it's not on the nucleus. Okay? Which type of molecules are utilized for this process? All of the RNA molecules, and including individual amino acids. So during translation, R, or ribosomal RNA, and really you could say ribosomes. Ribosomes are used, messenger RNA is used, transfer RNA is used, as well as obviously individual amino acids are used during translation. the end product is what? Proteins. According to translation, we can make proteins. We already identified the length of the protein varies depending on the code. When we say protein, it's super generic. This could be making RNA polymerase, which is a protein. This could be making the enzymes that carry out glycolysis, the Krebs cycle. This could be making proteins that cells use for receptors, like insulin receptor. This could be how your cells make insulin, your hair protein. You name it, almost everything's a protein that's functioning inside of a cell. It gets manufactured by this process of translation. What questions do you have at this point about some of these overviews of translation that I mentioned, we talked about? I want to go through and do some practice for transcription and translation. Okay, what I've just given you guys is a practice sheet to summarize transcription and translation. On the top of the table or the top of the handout, I have that complement relationship between DNA and as well as RNA and how they interrelate with one another. This is going to be something that you'll need to be familiar with. I'll just provide it for you for practice sake. But for exam-wise, you need to understand how these nucleotides are complement and how they bind and interact with one another. Below that is then what's described as called the genetic code. Let me see if I can find an image for you. Below that is what's called the genetic code, and this is just a key for all the amino acids. I am not going to hold you accountable for knowing the codons for all of the amino acids. When you go on to be a biochemist, sure, you want to know these things, but you're probably not going to go on to be a biochemist. You may, but at this point you're not. So tables, charts, no, this is good. If you look at it, it gives you a relationship between RNA nucleotides, and more specifically mRNA nucleotides, and the amino acid which they code for. And you'll notice that there's 4 columns and 4 rows subdivided into 4 rows within themselves. Okay, can you see how that's oriented? In the middle of each cell, you'll notice there's a name, like for example, phenylalanine. Okay, can you see that full name? That's the name of the amino acid. When you look in parentheses, there are the alternative shorthand notes for the amino acid. So I could write phenylalanine, I can't even say it, phenylalanine, or I could write PHE, or I could just write what? F. Okay? What about serine? What's the letter nomenclature for serine? Just letter S, or I could say SEL. Okay? You also can see the start codon. We can reinforce that. This mRNA sequence for a start is AUG. And if you notice, remember, this is completely unrelated, but I think about it when I see this sequence. You know, we just changed the place here of the tags, car tags at Oklahoma. We start out with A's, so you'll see a lot of AUG. So look for the AUG sequence of the tag. I don't know, you'll see some. Start codon. What are some examples of stop codons? What would be a sequence of a stop codon? UAA or UAG and UGA. So how this works is you on the far left column you'll represent the first nucleotide. You just line the sequence. On the across would be the second codon, and the far right would be the third codon. So it's kind of like your old multiplication tables like you used in grade school, but we're using letter code here. So I'm going to throw out a codon sequence, and I want to see if you can, don't shout it out, let's see if you can find it on your handout. So first letter is A. Second letter is A. And the third letter is G. What amino acid does AAG code for? Lysine. Very good. Do I have questions on how to read that table? No, I will provide. You will do this process, but I will give you the table. Okay. Additional questions?
I'm not sure if I read stuff.
Okay, so I'm going to pull these up here so we can see everything. Stretch or a question? All right. So if I want to read this, so I just gave out a sequence like A, A, G. Okay, so the way I would read it is my first nucleotide would come from this first kind of column right here. So my options would be U, C, A, or G. Okay? The second codons would come from this list. My arrows are going to kind of get mixed up, but that are highlighted in the yellow. U, C, A, G. Okay. The third options would come from the far right. Now, if I went with that first scenario of AAG, I would first come to, on this far left column, the area that correlates with A. So I would come to this chart here. Okay. Then I would move over to the column that correlates with A. So wherever these two intersect is the box that I'll be working with. Does that make sense so far? Yeah. In the third code, I just look at this far right. So I said G. Okay, so that means I need to look at the fourth line. And so I said AAG is a codon which I found, which tells me that codes for an amino acid called lysine. Additional questions? All right, so in front of you, you have a handout with the table, and I've given you a sequence, and you'll notice that the DNA sequence looks backwards because we're going to make our RNA sequence in correct order, so we need to read the template with the scribe kind of backwards. Okay, so you'll notice the DNA sequence is running 3' to 5' in its written form. And so your mRNA should be written how? What end should you write on the left-hand side of your paper? A 5'. Okay. So I want you to first transcribe your messenger RNA. Transcribe your, let me restate that properly. Transcribe your DNA into a messenger RNA sequence. On the bottom of this handout that I've just given you. When doing this, I would write large letters and just keep them right below the letter which you're trying to do the complement of.
This one that we're doing is the first three.
So I'm making mRNA. Which base should I not use? I should not use thymine or T. If I see a letter A in the DNA sequence, I should write a uracil or a U for the RNA.
So I'm not using the bottom.
Yes, you are. nucleotide is A, so my RNA would be uracil. Good, you. So the first process, if you need to, where you're writing the mRNA, just right between the DNA and the RNA, just write transcription. And that's what we're doing in this first process is transcription. If you have successfully transcribed your RNA sequence, then you can do what with it? Translate it. That's where you'll need the table. And you'll read it in left to right in. Hold on. Yeah, so for the amino acid sequence, use the 3-letter abbreviation. For methionine, just write MET. And for the stop codon, you can just write stop. And I do have a range of start and stop. Yeah, if you need to physically draw a box around 3 or a line between the codons, I would do that. You'll notice it's easy to have a translational error, which can happen inside of a cell. And you would misbuild your what? You try to misbuild your protein. Amino acid. In my handout, the abbreviation AA stands for amino acid, which is manufactured or brought in order to make our proteins during translation. Short on time. I want to conclude the next few minutes with picking up the extra credit work. Okay, but before I move on, this, you can keep practicing. You can make up your own generic sequences. Just your DNA nucleotide, just randomly pick something. It doesn't have to translate into a protein, right? As long as you get the practice of doing the complement and then using the table. Okay. Since the bulk of the class is not here, there are extra handouts. If you want more of this, you can pick this up on your way out. Okay. Also, I want to give you your handouts 8 and 9 to submit for next week, which will get ready for cell division. We will, on Thursday, we'll get into chapter 8, and so still, I've not had a chance to just lock myself in my office and produce those materials for you. So I'll give you your handouts. If I don't give them to you in advance, I'll print them out for you for class on Thursday. But I do have two graded handouts, handouts number 8 and 9. I'm going to ask that these be due number 8 due on next Tuesday and number 9 due on next Thursday. However, if you have a chance to complete number 8 before class tomorrow, it'll help you a ton. Okay? As I'm passing those out, if you want to submit your extra credit packet for your exam, you can have that ready to go. And let me pass it out and I'll address that real quick. 8 and 9,
Are you wanting the scan form back as well?
yes. Handout number 8 is due when? Tuesday, but it might behoove you to do it when? Before class, Thursday. And number 9 is due next Thursday.